back to Concepts of Biology. We'll continue to read Chapter 3, Section 3.3, Eukaryotic cells, ribosomes, mitochondria, and peroxisomes. By the end of this video, you will be able to summarize the functions of ribosomes, mitochondria, and peroxisomes. Ribosomes are the cellular structures responsible for protein synthesis. When viewed through an electron microscope, free ribosomes appear as either clusters or single tiny dots floating freely in the cytoplasm. Ribosomes may be attached to either the cytoplasmic side of the plasma membrane or the cytoplasmic side of the endoplasmic reticulum. Electron microscopy has shown that ribosomes consist of large and small subunits. Ribosomes are enzyme complexes that are responsible for protein synthesis. Because protein synthesis is essential for all cells, ribosomes are found in practically every cell, although they are smaller in prokaryotic cells. They are particularly abundant in immature red blood cells for the synthesis of hemoglobin, which functions in the transport of oxygen throughout the body. Mitochondria. Mitochondria, singular mitochondrion, are often called the powerhouses or energy factories of a cell because they are responsible for making adenosine triphosphate or ATP, the cell's main energy-carrying molecule. The formation of ATP, adenosine triphosphate, from the breakdown of glucose is known as cellular respiration. Mitochondria are oval-shaped, double-membrane organelles that have their own ribosomes and DNA. Each membrane is a phospholipid bilayer embedded with proteins. The inner layer has folds called cristae, which increase the surface area of the inner membrane. The area surrounded by the folds is called the mitochondrial matrix. The cristae and the matrix have different roles in cellular respiration. In keeping with our theme of form following function, it is important to point out that muscle cells have a very high concentration of mitochondria because muscle cells need a lot of energy to contract. Figure 3.14. This transmission electron micrograph shows a mitochondrion as viewed with an electron microscope. Notice the inner and outer membranes, the cristae, and the mitochondrial matrix. Peroxisomes. Peroxisomes are small, rounded organelles enclosed by single membranes. They carry out oxidation reactions that break down fatty acids and amino acids. They also detoxify many poisons that may enter the body. Alcohol is detoxified by peroxisomes in liver cells. A byproduct of these oxidation reactions is hydrogen peroxide, or H2O2, which is contained within the peroxisomes to prevent the chemical from causing damage to cellular components outside of the organelle. Hydrogen peroxide is safely broken down by peroxisomal enzymes into water and oxygen. This ends Chapter 3, Section 3.3, Ribosomes, Mitochondria, and Peroxisomes. In our next video, we'll read Section 3.3, Animal cells versus plant cells.